Welcome to Week 6 of Hazardous Waste Treatment and Disposal. This week we will summarize the concepts of hazardous waste toxicology, including the impacts to humans. Although hazardous wastes and materials may affect public health through corrosion, fire, leaks, or other physical processes, the most common concern is their toxicology. Toxicology is the study of poisons and their effects on living organisms. Knowledge of the principles of toxicology is important in hazardous waste management in order to assess the potential biological effects of hazardous chemicals and as a foundation for performing risk assessments. The field of toxicology is multidisciplinary and includes topics such as exposure routes, distribution of toxic substances within the organism, molecular mechanisms for toxicology, and movement of the chemicals out of the organism. The field of toxicology also includes a number of subdisciplines that cover a range of toxic materials. For example, toxicology can relate to food toxicology, natural products toxicology, and aquatic toxicology. Toxicity results from a number of dynamic processes, including absorption, distribution, metabolism of the parent compound, storage, and excretion. Toxic agents do not usually exhibit detrimental effects on the surface of the organism, but they proceed through a series of exposure and metabolic pathways in which toxic effects result when a chemical or its transformation products reach a site at a receptor molecule on a target organ at a sufficiently high concentration. When an organism is exposed to a hazardous compound, the material is often absorbed through three common routes. The routes are orally, through the skin or dermally, and through inhalation. The route of exposure is a function of the media to which the organism is exposed. For example, drinking contaminated water, breathing VOCs, or wearing clothing contaminated with pesticides. Once inside the body, the contaminants are distributed throughout the body via the bloodstream. In many cases, the chemical is detoxified through a biochemical mechanism. The outcome of most of these reactions is a more water-soluble compound that can then be more readily excreted. There are times when metabolic reactions can transform the compound to a more toxic product. Depending upon its characteristics and properties, the contaminant or its metabolites may bind to receptor molecules and provide the mechanism for toxicity. The frequently mentioned example is carbon monoxide binding to the receptor molecule hemoglobin, which results in toxicity because the hemoglobin is not able to carry oxygen to the body. To understand the mechanisms by which hazardous compounds are toxic to humans or other organisms, a good overview of the function of mammalian organ systems is necessary. Particular focus should be placed on the organ systems of the skin, respiratory tract, digestive tract, circulation, liver, and kidneys. The textbook will provide an abundance of information on these systems. Toxic effects vary substantially depending on the place where the compound is absorbed. For example, chromium is highly toxic when inhaled, but is not as toxic when ingested. The reason is that chromium is probably not absorbed as well in the gastrointestinal tract. The routes by which organisms are exposed to hazardous wastes are through natural processes in which the chemicals flow with the water, air, or food as it enters the body. As mentioned previously, the exposure routes are oral, dermal, and inhalation. After a compound is absorbed, it travels through the bloodstream. Toxic chemicals rarely exhibit localized effects at the point where they are absorbed, but move through the body where they may attach to receptor molecules and then exert toxic effects. Common organs where binding occurs and toxic effects are realized include the liver, the kidneys, and the blood. Storage may or may not occur depending on the partitioning behavior between the contaminant and the different molecules, including fat. Chemicals or their transformation products that are very water soluble and do not have a propensity to partition into fats are rapidly eliminated in the urine. Organic chemicals with high partition coefficients may retain in fat for years or decades. Metals may also be stored by binding to fat and other biological molecules such as proteins. For example, lead binds with the molecules of the central nervous system, and cadmium binds to molecules in the kidney, causing kidney damage. Cancer is a disease in which altered cells divide uncontrollably, resulting in tumors. Most chemically induced cancers are characterized by latency periods of up to 40 years, a phenomenon which really complicates cause and effect determinations. In general, cancers that result from exposure to hazardous wastes are caused by chemically induced mutations. The ability to quantify the toxicity of hazardous chemicals is essential in hazardous waste management. Industrial personnel and the general public can be exposed to hazardous waste as a result of chemical spills, consuming contaminated drinking water, or breathing fugitive air emissions. Therefore, it is important to have some ability to quantify toxicity. This quantification of toxicity has resulted in values that describe acute exposures and chronic exposures, short-term and long-term exposures. For example, the LD50, the lethal dose to 50% of the population, is used to quantify acute toxicity, whereas the threshold limit value, TLV, has been developed as a guideline for lifetime exposure to chemicals. Some of the systems used in quantifying toxic responses have been developed for specific environmental programs. For example, the maximum contaminant level, MCL, is used in regulating drinking water under the Safe Drinking Water Act. The most important factor that influences toxicity is the dose. A second important factor is the time period of exposure. Toxicity is also often classified by the number of exposures or the duration of exposures. Related exposures may be classified as acute, subchronic, and chronic. In classifying toxic action based on developmental effects, the life stage at which toxicity occurs is the basis for classification. This life stage could range from fertilization to adulthood.